Hi again. So we will continue lecture number 2 about research databases, and now protein databases. Remember that on molecular biology you have a dogma that starts with a gene that is located in the genome, that is transcribed into mRNA, that you can have on databases, on DNA databases in the aspect of cDNA, and then that mRNA in the dogma of molecular biology is converted into proteins. So there are also research databases of proteins. Along the time, there have been a growing number of entries on proteins. The main protein database is UniProt, SwissProt, that nowadays it's only UniProt. That database has the sequences, all the sequences of proteins annotated. As we have been talking, we have the example of Tata box binding protein, human Tata box binding protein. In the UniProt database, it has a unique session number that is P20226, and the entry name is called TBP human. Now, the display of UniProt database is based on function, names and taxonomy relative to the organism, the functions and localization of that protein, and it has a full left menu with the sequence, structure, family and domains, all the annotations. The last kind of annotation that was included is gene ontology, that is based on molecular function, cellular component, and a cellular process where the protein is involved. According to UniProt, the type of data presentation is this one, and it allows you to do the blast immediately, add to your profile this protein in order to study more, and this database, it's the European database, but as for the other databases, it has also a database from USA, the NCBI database, that is called GenePep, the specific database for proteins. And once again, for this you have its accession number and all the information from the organism, the references, and if you want the FASTA format of this protein, you only access to FASTA. This is the GenePep display of the protein in this GenePep database. It has also the presentation, graphical presentation, where you have the annotation of several domains and features, conserved domains, and you may follow these links in order to know exactly which features are, and these are transcription factor interaction surfaces that are located along the Tata box binding protein. Also is available the protein 3D structure. This kind of structure is available on PDB. It's a protein data bank. It's specific for 3D structures of proteins. Now you can see the example of this protein from the gene until the protein. And at UniProt, where you can have access to all the proteins, and this is a combined database from SwissProt, UniProt, and you can follow and check all the characteristics of this protein. You have the name as TPB, as the same name as the gene. It is located on nucleus, so the gene ontology identifies this protein as belonging to nucleoplasm. It is related to some kind of diseases, and it has a chain that is responsible for Tata box binding protein. And Tata box is a sequence in the genes. Along the UniProt annotation of the protein, you have always this structure section. Secondary structure, you may have access to it and check the locations of beta strand and helix. These structures may be predicted using PsyPred. Is one of the tools that allows you to predict secondary structures. 3D structures are very complex, and they may be accessed in PDB, so Protein Data Bank, and you may check it and have a view using JALView and have a 3D profile that allows you to see the several helix in this protein, and most of the times you have different kind of views according to the perspective and also according to the main components. In this, you can see the double helix of DNA combined with the protein that has the beta strands and the helix that are the secondary, and all these turns that become the 3D structure of all the protein that can interact with the molecule of the DNA presented in blue. Here the other strand of DNA, emphasis in here, one part of the protein, one subunit, and the interaction with the DNA. So, as I was telling you, Tata box, it's a segment of a strand of DNA that is connected to this protein in order to perform the transcription of a gene. Now, in different colors according to some of different perspectives of this same protein. About proteins, you may also find at the UniProt and at GenePep some links to other databases, and you have cross references from this primary database into other proteins. For example, protein at UniProt exactly from the database of NCBI. So, although this is a primary database, it has already some cross-references to other databases. But there are specific integrated databases that integrate information from different primary databases. So our next talk will be about integrated databases. Thanks for your attention. Meet you at the next video about integrated databases.